Hello, everyone. I am Samuel Okyem, and my advisor is Jonathan Swidler, and my co-advisor in the MBA program is Martin Gillette. Today, I'm going to talk about the discovery of neuropeptide isomers in the central nervous system of mammals. So starting with what are neuropeptides? So these are very diverse molecules that are very important physiological effects and also some behavioral effects. Mostly the peptides are produced from precursors or propeptides that need to undergo several post-translational modifications to get an active form of peptide. Now, one of those post-translational modifications that these peptides undergo is the conversion of one of the amino acids from an L form to a D form. And it happens that this is not likely detectable because the state-of-art equipment that is being used for peptidomics, which is very high throughput and very sensitive, is mass spectrometry. And since there is no mass shift, then it becomes very hard for you to differentiate between these two isomers because you don't have any mass differences. So although mass spectrometry is not able to distinguish between these two peptides, but it's still a very sensitive and a very high throughput procedure. So we can still harness that activity by couple of it other kind of techniques to actually identify these peptides. So in our group, we have different ways that we couple mass spectrometry and also some other techniques. So one of them is ion mobility, where you look at the gas phase structures of peptides, look at their mobility in a gaseous buffer. And then if these peptide are isomers, sometimes with a very highly sensitive ion mobility instrument, you can resolve these two different isomers. Another way is looking at their activity towards proteases, that is the ability for them to be susceptible to protease degradation. So in our group, we observed that some of these isomers or the amino acid containing peptide may be resistant to protease activity. So we kind of use that in our assay as well. So there's a couple of these procedures that we employ in our lab. And using these procedures, we've come up with some of these peptides in several organisms. So as you can see, from lower organism planarians and then other metazoans, we have discovered several of these, very few, but some of these, the amino acid containing peptides. And now the question I'm trying to answer here is that, does this thing actually cut across all species or organisms? And can I find some of these, the amino acid containing peptides in mammals? So first, I'm going to employ some of the techniques that I talk about. That is liquid chromatography, ion mobility, mass spectrometry. And I try to go into the hypothalamus of rats because this is where you can find a lot of these endogenous neuropeptides. And by using this approach, that is liquid chromatography, ion mobility, mass spectrometry, I was able to discover tons of peptides in the hypothalamus of rats. And of course, some of these are proteins that degraded in the course of the analysis. So I have like 5,000 peptides that are actually discovered from these at run or analysis. So this is about a lot of peptides to know which one of them is undergoing isomerization. So therefore, I have to come up with an approach. So based on our previous data analysis in our group, there are some of the physical chemical properties that we actually use to identify or discover some of these peptides. And one of them is the retention type differences. So you can have a peptide that is coming off at two different retention types. So the same peptide, you can see it at two different retention types. And that's one of the criteria that you can use to try and identify some of these peptides. In our group team, we discovered that sometimes some of these peptides may have different fragment ion ratios. So the fragment ion ratio distribution of these peptides coming off at these two different retention times may not be the same, and they may be different. Another factor is the ion mobility. That is the mobilogram in a gas phase. That is how they move in a gas phase may be different for the two peptides occurring at these two retention times. And the last one is the protease activity that I discussed previously. So using all these procedures, how do we come up with, combine all these procedures to discover new peptide from these 5,000 peptides that I discovered? So you can see here that employing all these techniques, we found 252 peptides to actually be part of our criteria that we use in our data analysis approach. And now we further said that, oh, this 252 peptides is actually a lot, and we still cannot tailor it down. So we just resorted to the protease activity, which has been one of the significant ways of trying to discover these neuropeptides. So we try to see which one of these 252 peptides is resistant to the protease action. And then we found out that 42 of them is actually resistant. And among these 42, which one of them is neuropeptides? So from there, we came up with this table. I mean, I did not include all of them. Some of these peptides are also fragments of the actual endogenous peptide that were discovered, not the entire sequence. So looking at this table, there is a couple of them. We have copeptine fragments, we have cereblin, we have substance P, neuropeptide K, galanin, and we have cocaine and amphetamine regulatory transcripts. So some of these peptides may be potentially undergoing, some of the amino acids may be undergoing isomerization. They may have a D-amino acid containing peptide isomer. So then I pick up galanin from here because galanin is a very, very important neuropeptide. It's been implicated in feeding, behavioral responses, even in diabetes in rats. So it's a very essential peptide to look at to see if there is an isomeric form or a diastereomeric form of these peptides. So looking at galanin in details, now you can see here that indeed galanin is coming out at 2 retention times. There's a second peak of galanin, which is very persistent and very predominant here. So you can look at the MS-MS spectra, and you can see that in fact they have correlating MS-MS spectra, and indeed galanin is having coming off at 2 retention times. Now the question is that does these two galanin have the same ion mobility? Now you can see that the first galanin peak has different ion mobility compared to the second galanin peak, which is an indication that there may be a possible isomeric form of galanin. Now the question is that is this observation, that is the 2 retention time observation, coming from our approaches of extraction or other things that we are actually using in our procedure trying to identify this peptide? So what I did is to get a synthetic galanin, spike it in the tissue homogeneous, take it through the extraction process, and see if I'm going to observe a second peak. And you can see that I cannot see the second peak, which means that possibly the second peak is actually endogenous. It's actually found in the tissue that I was trying to analyze. So further on, I went on to get fractions of galanin itself and try to see if indeed a fraction is also resistant to protease activity. And you can see from here that the fraction is somewhat getting reduced. Once we apply the proteases and try to degrade them, it gets down. But then there is an observation of this peak coming up. That is, the truncated form of the peptide is actually found. It looks like the enzyme is getting locked here, which we don't know what is happening and why we are observing this. But so this is a clear indication that the peptide is somewhat resistant to the protease activity. Now, the challenge also here is that there is a couple of, when you look at galanin sequence itself, we go back. Look at the sequence itself, you can see that there is a aspartate and asparagine. So there is a possibility of isoaspartate formation in this particular peptide. So the question is that is the isoaspartate also present, or is it there's a second peak as a result of isoaspartate formation? So what we use is an endogenous enzyme, which is protein isoaspartate transferase, and this enzyme can actually carboxylate the isoaspartate in peptides and then convert it back to an aspartate. So it's like a correction enzyme that correct isoaspartate in peptides endogenously in organisms. So we try to harness the activity of these enzymes and try to see if we can correct the observation that we are seeing. So we can see that the second peak actually disappears when we treat the extract with cream T, which is protein isoaspartate methyltransferase. We are seeing a very small amount of the methylated form of the peptide, but it's not that prominent. So there is also a couple of aspartate and asparagine residues that can actually lead to the formation of isoaspartate. So the question is, which one of these residues may be undergoing isomerization? So I kind of treated my fractions containing galanin with chymotrypsin and try to see which one of these fragments will be coming up at different retention time. You can see that this particular fragment containing asparagine is actually coming up at 2 retention time when you compare with the standard. And then you move further with ion mobility profiles, and you can see that they have slightly different ion mobility profiles. You also move on to look at this second peak, and you can see that the second peak, which contains aspartate and asparagine as well, has 2 retention times. But the ion mobility profile is not that different. It's hard to tell whether these two is the same or not, but we're still investigating that now. The last work that I'm trying to do this summer, and a couple of work that I'll subsequently do, is also try to use the same enzyme, but this time around, I'm going to induce a mass shift. So it happens that when this peptide actually transfers a methyl group to the carboxylate side chain of the isoaspartate, there's a formation of a cinnamide intermediate. In the presence of excess trace, the amide of the trace, the amine of the trace can actually attack this the carbonyl and then you can have a mass shift of about 103 Dalton. So looking at this mass, when this mass shift is observed, then it can be conclusive that indeed your peptide contains an isoaspartate. So this is what I'm working on currently. I'm also looking at trying to isolate galanin itself, the different forms of galanin, and characterize it, uses matrix reagent to actually understand the amino acid in there and whether or not they are undergoing isomerization. So I would like to thank my advisor, Jonathan Swidler, the MBM program, and the entire Swidler group for this. Thank you.
Any questions?
I know I'm going to hear more about this at your call.
Yeah.
It's coming up in a couple weeks. But was it known that there was galanin you're in the pituitary? Right?
So
Are any of these novels finding them in the pituitary? That's my question, really.
So I think that it's been reported that it's found in the pituitary, but I analyzed the pituitary myself, but I've not done it a couple of times. I think I analyzed it twice and I couldn't detect it there within my samples. So although in literature it's reported that there is galanin in the pituitary, I did not find it myself, but I still have pituitary samples. I can do more analysis, but since I found in the hypothalamus, I kind of focus on that.
Oh, okay.
Yeah, but it's been known there, but I didn't find it myself.
So when you say hypothalamus, is that the whole hypothalamus or a region of the hypothalamus? Because that's a very peptidogically rich area.
Yeah, I think it's the tissue is the whole hypothalamus.
Thank you, Sandy.